Hello everyone and welcome to Prof Talks. In this presentation, we will review the USFDA 483 observations that were issued for the Ipca Ratlam facility, which is an API manufacturing facility for the inspection conducted in June 2023. In this presentation, we'll try to have a look at what are these 483 observations and what we can learn from them to avoid similar kind of observation within our own domain. The redacted copy of the 483 observation is publicly available, and USFDA has taken due care to prevent all the confidential information. And hence, we have a disclaimer that the sole purpose of this presentation is to gain knowledge and training onto the learning from this 483 observation. We do not intend to take any advantage of the name of the organization or any due disrespect for any of the observations cited. The sole purpose of this presentation is learning. So if you can see, the inspection was carried out from 5th June 2023 to 13th June 2023, which is 7 working days. It was carried out by 3 inspectors, which are called as investigators, Mr. Rajiv Srivastav, Salim Akhtar, and Menzeng Zhang. And this is an 18-page document which cites 11 483 observations. So the US FDA issues this 483 report at the end day of the inspection. Other regulatory authorities issues this observation report some due days after when they reach their head office, that is 15 days or something. But US FDA prints and submit the signed copy to the site team immediately at the end of the inspection, and the time cycle for responding starts from that very day. So this is a very big important difference between US FDA and the other organizations. Before moving ahead, please ensure that you have subscribed to the channel Prof Talks and have pressed the bell icon so that you can receive the notification of the upcoming presentations. As we have seen in the previous slide, US FDA has cited 11 483 observation in this particular 483 report. US FDA categorizes these observations into one umbrella heading and reports few of them into one observation. For example, if you can see, the first observation cited here is investigations are inadequate to determine the root cause for failure of the batch, which includes the OS, deviation, and market complaints. So there are total 6 observations cited under this one umbrella observation. 2 examples each for OS investigation, 2 for deviation, and 2 for market complaints. The second observation is quality unit failed to take adequate action against failing API batches sold in the market. So this specifically is related to product recall and which has only one observation under it. The third observation is reworked API is not subjected to appropriate evaluation, testing, and stability testing. Again, one observation only. Failed to establish optimal processing condition to ensure consistency in critical quality attributes of API. Again, a single observation, the fourth 483. The fifth one is firm failed to oversee and control over manufacturing of drugs to ensure quality and managing risk of establishing safety of materials used in manufacturing. So they have cited 2 observations under this heading. The sixth 483 is firm failed to ensure all specifications and test procedures are scientifically sound and appropriate to ensure raw material and API conform to the established quality standards. 2 observations under this category. The seventh 483 is laboratory tests conducted to determine conformance to specifications for each batches are deficient. So 2 observations for that. The eighth one is quality unit failed to ensure all deviations are reported onto the batch certifications that are certified by the third party consultants. So there are 4 observations in this. The ninth one is calibration of laboratory equipment in the microbiology laboratory is deficient. So they have cited the example of pH meter here. The 10th one, determinations made to establish suitability of secondary standards by comparing against primary reference standard is deficient. Again, a single observation. And the last one, 11th, is failed to maintain the complete records of major equipment use, cleaning, and maintenance. Now we go into the details of the observation. So the first observation, as we have seen in the summary, is like investigations are inadequate to determine the root cause for failure of the batches. So there are two examples each for OS, deviation and market complaints. So the first example for the out of specification investigation was like a lab recorded OS result for API for assay test by HPLC method. The OS result was reported for 2 out of 3 batches that were analyzed in the same sequence. Okay, so in phase 1 investigation, it was determined that air bubbles in some solution or something was the root cause. This root cause was proved through hypothesis test used only for one of the failing batches. Then both the OS batches were retested based on the hypothesis testing, and the assay test results was passed. Initial invalidated, the OS result was invalidated. So the problem is the root cause is not fully supported by the hypothesis test, such that hypothesis test does not include both the failing batches. So this is one of the major problem. Like the hypothesis testing which was done, it included only one out of the 2 batches and not both of the batches. In addition, the graph, some graph and the percent RSD is not supporting the root cause. So the root cause is not supported by some statistical data. Okay. And the third party quality audit system consultant flagged this OS result investigation. So escalation. So the data and investigations are being reviewed by the third party consultants, and they flagged this investigation. Firm already submitted a remediation plan and to this consultant. However, did not have any feedback from the quality consultant to verify whether the remediation plan was adequate. So there are two issues. The hypothesis testing was not supported by testing both of the batches, and they do not have a feedback mechanism to ensure that the plan that what they have proposed is adequate. Moving to the second observation for the OS investigation under observation 1. So a lab recorded OS for API for solubility test in some solution. The OS result was reported for 2 batches. The phase 1 investigation did not reveal any assignable root cause, and the study proceeded to phase 2 investigation. The report stated that no assignable cause was identified in the phase 2 manufacturing investigation. However, this information is not available in the investigation report. So this is an important point, that no assignable cause was identified in the phase 2 manufacturing investigation also, but this was not clearly demarcated into the investigation report. Okay, the investigation continued into the phase 2 laboratory investigation and determined lower temperature of some solution or something as the root cause for a hazy solution, and undissolved particles settled onto the bottom of the test tube. New samples were prepared and retested, and based on the passing results, the initial OS results was invalidated. So the problem here cited is the root cause is not supported by the hypothesis test. Okay. In the previous example, we have seen in the previous observation that the problem was out of the 2 batches, hypothesis testing was done only for 1 batches. And in this case, no hypothesis testing was done. Okay. The solvent is stored at room temperature or some specified temperature, and the laboratory is maintained at not more than some specified temperature. And again, similar case, the third party quality consultants have flagged this. The firm submitted some remediation plan, but they did not have any feedback mechanism to ensure that remediation plan was adequate. Under the first 483, moving to the two observations related to the deviation investigation. So the first one, the firm recorded a deviation for damage of a machine during manufacturing of the API batch, which was implicated to a bolt for damaging some part. The investigation found that the bolt was dislodged from the position. The investigation confirmed that a nut was missing, and it was assumed that the nut was drained out with water during partial cleaning. The investigation does not include a picture to verify all parts involved in assembling and to verify that all the parts were accounted. So this is mainly related to the proof associated with the investigation. So the firm did not attach any photographs to ensure that all the parts that were of the machine were accounted previously and after the processing. And second observation is also of the similar category. The firm recorded a deviation for damage of the machine during the API batch and implicated a screw for damaging. The investigation found a missing screw from the SOP display board. Okay, so it was assumed that the missing screw of the display board fell inside the material during unloading and which caused damage. In this case, again, there was no photograph included for the SOP display board and the surroundings to assess the likelihood of falling of the screw in the materials during the unloading process. Moving to the two observations related to the market complaint investigation. So the firm recorded a market complaint for foreign particle including metal-like particles and magnetic particles in the API powder inside the inner bag of a non-US market batch. The investigation did not find any assignable root cause. Okay. The firm uses some machine to control the extraneous matter and the magnetic particles in the API. However, the firm has not qualified that machine. So this is a major point, that the firm uses a machine to control the particles, the magnetic particles and the extraneous particle, but they have not been qualified, and also they do not have the challenge test before, during and after the preparation. So a challenge test is not in place for that machine to ensure that it is functioning properly. Okay. The second observation is the firm recorded a market complaint for foreign particles with metallic appearance in some non-US batch. The investigation did not confirm the nature of the black particle, whether it is a metallic or non-metal. So the investigation was carried out, but they did not ensure whether the particles are metallic or non-metallic. The investigation stated that some machine is not contributing factor for the foreign particles. However, they did not include all the manufacturing equipment to ensure that there is no inclusion of that particles into the machine. And this has been supported, like they recorded a deviation for black particle in an API batch, and the investigation implicated damage of some machine for that API particles. So this is supporting the fact that they have not included all the other machines to ensure that there is no black particle generation from other machines also. So what are the key takeaways from 4831 related to the investigations? Root cause identified during OS investigation should be supported by hypothesis testing before invalidating the OS results. Any hypothesis testing done during the OS investigation should consider the hypothesis for all the failing batches. Any communication escalation related to investigation should have a feedback mechanism, and same should be captured into the investigation report, like in the case of the consultant where the firm has given some remediation plan but didn't included the feedback from the consultant. Conclusion of phase 1 and phase 2 investigation, like no assignable cause identified, should be clearly mentioned in the investigation report. Pictures wherever necessary should be captured in the investigation as a part of a supporting evidence. Metal detection machines deployed to control metal contamination should have a qualification, and there should be some checks before and after an in-use challenge test to ensure that the machines are operating properly. If some complaint of metal detection is received, documented evidence should include the investigation to conclude or rule out the metal possibility. Moving to the second of the 11 observations, the quality unit failed to take adequate action against failing API batches sold in the market. So this is specifically related to the product recall. The quality unit failed to take adequate actions, including product recall against batches of API that failed to meet the impurity specification during stability testing. The lab initiated an OS result for two batches that failed to meet the impurity specification for a specified impurity, any unspecified impurity, and total impurities. So a table has been presented which has been redacted, but there has been an OS into a specific impurity, any unspecified impurity, and total impurities. Subsequent lab testing confirmed the original OS result. During manufacturing investigation, no assignable root cause was identified. However, the manufacturing investigation concluded the batch failing might have been due to the high concentration of some XYZ that caused the increased value of the impurities. And to mitigate that risk, a CAPA was initiated to change the washing quantity of water from ABC to suppose PQR. Okay, both the batches were consumed in the domestic market. For impacted batches, the quality unit failed to take action, like considering the market notification to the regulatory authority and notification to the customers. As per the PQR, the firm has manufactured many batches that were used for domestic and international market. Out of the marketed batches, the firm tested retained sample of only a few batches and not all the batches. So what we can learn from this observation second related to product recall is the quality unit should have a risk-based mechanism to evaluate the requirement for some market action like recall, notification to the customers and notification to the regulatory authorities. Many of the big pharma organizations use a quality matrix or a risk matrix to evaluate the need of first the escalation to the management. So there are some specific criterias like impact onto the product quality, impact onto the patient safety, impact onto the company image, and if that risk comes out to be high, then that particular situation is escalated to the management, and there is a specific committee at the management level which takes a decision for like notification to the customers, notification to the authority, or initiating a recall. So this can be a better approach where initially risk is evaluated at the site level, escalated to the management, and the decision is taken by the management for further action. Okay, to mitigate the risk of any impact on already marketed batches, the retained sample of all the batches should be tested and not just the few batches. So for example, there are 10 batches in the market. So to mitigate the risk of those 10 batches, you have to test the retained sample of all the batches to specifically ensure that there is no impact on the quality of that batches. So moving to the third observation, reworked API is not subjected to appropriate evaluation, testing, stability testing, and documentation to show that reworked product is of the equivalent quality to that produced by the original process. Okay, so the firm did not follow the SOP for reprocessing and rework of the API. So this is the first observation in itself, that the firm did not follow the procedure. So this is a deviation in the first statement itself. And how this procedure was not followed, that you released multiple reworked API batches without satisfactory evaluation of data, including 6 months of the stability data. So 6 months of the stability data was to be evaluated to release the reworked batches into the market. There were few batches that were released into the market, but stability data was not evaluated and did not place the aforementioned batches onto the stability study. So what is the key takeaway? First and the basic requirement of CGMP is you have to always follow your SOPs and you did not deviate. So whatever your SOP says, you have to always do that. In this case, if the SOP stated to have a 6-month stability evaluation, the firm should have followed the 6-month stability evaluation before the release of the batches. And second, if reworked batches should be evaluated and supported by stability data wherever required. So if the firm has established some criteria on a risk-based approach or some basic traditional data, that some specific criteria of evaluation is needed for reworked batches, that specific criteria should always be followed. Moving to the fourth observation, the firm failed to establish optimal processing condition that ensures consistency in the critical quality attributes, that is CQAs of the API. The validation production record for API process performance qualification instruct for the process as the details have been redacted, and there is no instruction on how much material should be loaded and how much and how to perform some activity. So there is no specific clarity into the manufacturing process during the process performance qualification. Okay, during the walkthrough into the manufacturing building, the investigator noted that the material and there were big as 2-3 inches. So this is the information has been redacted specifically. However, the main concern here was the general manager of production have confirmed that the validation was carried out in 2018, and this was the only validation that the firm has available. In year 21 and 22, there are many OOS recorded and no due consideration have been given to revalidation. So what we can learn from this 483 observation is like process validation should ensure appropriate processing steps with clear understanding in how much and how to carry out the activity for each step, and which should ensure the critical quality attributes of a particular process. Okay, and due consideration should be given to the periodic revalidation to ensure a state of control. The fifth observation, firm failed to oversee and control over manufacture of drugs to ensure quality, managing risk of and establishing safety of materials used in manufacturing. So the firm uses some XYZ as the raw material to manufacture the critical starting materials of the drug substance. The firm tested batches of the starting material that were used to manufacture the batches of the drug substance. Among the batches, of dash the test results of the batches were in the range of this. However, the firm did not conduct the proper contamination testing in the drug substance, and also they were not aware of the potential hazards if this testing was not done. So the firm distributed XYZ number of batches into the US market. In addition, retest date of the batches will be due in some specific time. So as per the completion of this investigation, the firm has not initiated any plan for risk assessment for this HCQS. Moving to the second observation in the fifth 483, the firm's standard operating procedure for complete cleaning of equipment used for the processing of API requires a complete cleaning of various equipment used to manufacture the drug substance during the production campaign. Okay, this SOP defines the frequency to conduct a complete cleaning of the equipment train shall be some specific period which comes earlier. So basically, if we understand this, so there is some basic contradiction within the SOP itself related to the cleaning frequency of the complete equipment chain during the production campaign and at the end of the production campaign. Also, the carryover study did not provide scientific justification that the commercial manufacturing process does not carry over from the starting material. The key takeaway which you can take from observation 5 is first of all, we should understand that cleaning validation itself is a very vast topic, and there has been numerous studies and numerous thoughts and documents available onto the cleaning validation aspects, and there are many cleaning validation experts available into the market, into each of the industry. They have their own expertise into that area, so the firm should utilize all those expertise in understanding such kind of observation and implementing more enhanced strategy. But specific to this, a risk-based contamination study should be available wherever there is a potential risk of contamination through key starting materials and raw materials. And a risk-based cleaning strategy with adequate cleaning frequency and potential for carryover need to be available for all the products. Moving to the 6th out of 11th 483s, firm failed to ensure that all specifications and test procedures are scientifically sound and appropriate to ensure that raw materials and API conform to the established quality standards. Okay. The firm conducted 2 carryover studies to justify the commercial manufacturing process does not carry over from the starting material to the drug substance. So the firm's study report was designed to spike some percentage into the starting material of the drug substance, and the drug substance was produced at the lab scale with a spiked starting material. Okay. However, the firm's commercial batch size for the drug substance is surely some X scales larger than the lab scale, and the firm did not provide any justification to provide a comparison between the lab scale batches and the commercial scale batch. So comparability study between the lab scale batch and commercial scale batch for ensuring that this spiking study is applicable at the commercial stage also is not available. And the second example is like the firm carryover study was designed to test some XYZ component into the drug substance from commercial scale batches, and which was found not detected. The firm did not test that material into the starting material used to manufacture the commercial batches. The firm did not provide any scientific justification to provide that the level of starting material does not impact its carryover into the drug substance. Moving to the second observation under 6th 483, the firm's specification and analytical procedures uses GC method to detect related substance and quantitate total impurities in the critical starting material. So instead, what the firm does is, instead of using an independent assay test, it deducts the total impurity percentage from 100% to calculate the percent purity, that is assay. So the firm did not provide any scientific justification or conduct any sound study to prove that deducting total impurities percentage from 100% to calculate the purity percentage, that is assay, is equivalent to an independent assay test. Okay, so here a scientific rationale, a justification, or some study report is not available for the approach that has been used by the organization. Okay, and the second aspect in this is, instead of 6 standard injections performed into your method validation during system suitability, your firm executes 3 standard injections in the test method for system suitability test. So the method validation uses 6 injections for system suitability. However, in the routine use since 2018, the firm has been using only 3 injections for system suitability, which is not in line with the method validation, and hence it puts the question onto the method that has been used. So what we can learn from the sixth 483 in overall is, any studies conducted at lab scale should demonstrate equivalence for the commercial scale batch size with adequate scientific justification. Okay. Consideration should be given to ensure that there is no carryover of any potential impurity from starting material in the drug substance, and it should be done either by testing or through a scientific rationale. Okay. Any analytical test reporting adopted which is being used a different approach than the routine approach should be supported by a sound scientific rationale. For example, in the case of 483, where SA was calculated by subtracting the percentage of total impurities from 100, so there was a different approach than the traditional SA method approach. So there should be a scientific rationale why this approach has been adopted and why this is acceptable, and methods that have been adopted during process or method validation should be used as such with no change without proper justification. Okay, so in this case we have seen the method validation for system suitability, 6 injections were required but only 3 were being used in the routine testing, so there was no justification for it, and hence which put the method used into question. So such things should not be done. Moving to the seventh point, laboratory tests conducted to determine conformance to specification for each batch are deficient. Specifically, laboratory tests conducted to determine multiple batches to the return specifications are deficient, as deficiencies were observed in the processing methods and integration of peaks during analysis of impurities and residual solvents. So the problem here is the QC lab uses a different processing method with inconsistent peak integration parameters to integrate peaks of standard and different lots of a product into the same HPLC run. Here an example has been cited where QC lab tested 2 batches. The lab uses 3 different processing methods to integrate peaks. Okay, so of the lots. So peak integration parameters used for the batches are given into the table below. Some uses like a value to value approach or a force peak or set minimum area. Different methods have been used into the same HPLC run. And under these integration parameters, any impurity with a peak area of less than some integrated value will not be eluted and hence will not account for the total impurity. Similar kind of observation is cited under the second observation, where a lab tested 2 US batches for residual solvent by GC and used 3 different methods to integrate peak of standards and lots, and same table has been provided where value to value or set minimum area of force peak have been used, and under these processing parameters, any potential unknown peaks with peak area less than a specific value for a specific time frame will not be integrated. So the learning we could have from the 4837 is, like, due consideration should be given to peak integration parameters. As we have seen into the some previous 483s issued by USFDA, due consideration has been given to the peak integration parameters, as they can mask the test results and they can give some false results also. So the parameters that are used for peak integration should be very considerate and should be harmonized across for the integration of the peaks. The eighth observation state that quality unit failed to ensure all deviations are reported onto the batch certification certified by the third party consultants. Okay. The firm's quality unit failed to ensure that all deviations generated during the manufacturing of the shipped batches were reported by the certifying organization, that is the third party consultant. So the firm shipped some batches during the COVID period to the US. The third party consultant failed to document significant deviations observed during the manufacturing onto the batch certificate. For example, 4 US batches failed to meet the yield specification. However, the third party consultant failed to mention these critical deviations onto the batch releasing certificate. So this is one of the major thing, and additionally for this 4 deviations of yield, the firm conducted one umbrella investigation. So if it is observed in 4 different batches, it is expected that different deviations should be logged and investigated independently, but a firm investigated under one umbrella investigation. It was discovered during the investigation that this umbrella investigation was not initiated into the firm's quality management system. So it was like the firm was using trackwise, but this investigation for the 4 batches, it is not a part of the quality management system, which means that it is some parallel investigation ongoing, which is not documented into the quality management system of the organization. The other 3 observations under the 8th 483, like during release testing of a batch for residual solvent by GC, an extraneous peak was observed at some RT. The peak was observed in all 3 batches, and the firm initiated the deviation, but the third party consultant did not document this deviation onto the batch certificate. The C observation is of the similar kind for residual solvent analysis by GC. Again, an extraneous peak was identified, which was attributed to some laboratory environment, and this was again not included into this deviation was again not included into the batch certificate by the third party consultant. And the D observation, that is specific, like there was no documented evidence available during the inspection. The third party consultant reviewed system audit trails and product audit trails pertaining to the overall assessment of the projects and systems used to certify the batch. Also, the protocol used to certify batches did not specifically ensure the review of duplicate sample injections and control and test injections, etc. So what do we learn from this 483 is like critical deviations during the manufacturing should be recorded onto the batch release certificate whenever there is such a requirement. Deviations impacting multiple batches should be investigated separately, and all the deviations and any other aspects or events or incidents should be recorded within the firm's quality management system, which is a requirement of ICH Q10 as well. And any activity carried out, that is audit trail review, in this observation should be documented into the GMP system. Review of the document should be comprehensive to cover all the associated details and the aspects. The ninth 483 states that calibration of laboratory equipment in micro lab is deficient, and this was specifically for the verification of the pH meter. So the verification of this pH meter is performed as per SOP using specific pHs with slope acceptance criteria from some X percentage to Y percentage, and a table has been given. So the micro lab considered the slope values of some percentage which is generated for not all the pH values but for some specific pH values only. So the microlab operator who routinely uses this pH meter also stated and confirmed that he looks for the slope for the specific, for example, if 5 specific pH values are given, so he considers the value for only 3 specific pH values, slope value only, and if the other 2 values are out of range also, that values are not considered and consideration is not given for any investigation or the equipment performance if the values are out of the slope is out of value also. Moving to the 10th 483, the second last one, determinations made to establish suitability of secondary standard by comparing against primary reference standard is deficient. So the in-house qualification is carried out for some secondary reference standard, and this is carried out by 3 replicate analysis assay samples for the API, okay, are prepared and compared against the primary standard. As per the SOP, the qualification SOP, the percent RSD of triplicate assay should not be more than some specific XYZ percentage. However, the site does not have any scientific rationale for this percent RSD criteria. For example, if you state some RSD criteria is there and you have to meet that RSD criteria for comparing the 3 replicate assay samples for the secondary standard against the comparison with primary standards, but what is the origin of that percent RSD and how you have arrived to that percent RSD is not available. So the key learning what we can take in a generalized manner from here is like every decision taking aspect should have a sound scientific rationale. So whatever thing you do, whatever thing, if there is an origin of something and you are taking a decision onto that aspect, so you should be very sure that what is the origin and what is the rationale why we are using. So we should develop a habit of questioning why we are doing this and how this thing has arrived, right? So coming to the final 483 number 11, firm failed to maintain complete record of major equipments use, cleaning and maintenance. So this is one of the very basic observation, like partial page and information are missing on page 1 and 2 of the equipment cleaning and usage maintenance record in 2020 for some specific equipment, which is a major equipment used in the manufacturing of the drug substance. So the logbook, the partial page or some information is missing from page 1 and 2. Okay, so what we have to learn from is this, like we should always ensure good documentation practices and data integrity principles. We are thorough with our Alcoa plus principles of complete and available records. So the USFDA does not emphasize this observation as to some data integrity principles or something, but if you can see, the information is missing into this case. Like, so it is very important that all the information, all the GMP information that we have as a part of a batch record or any other documentation associated with the batch should be always available within the quality management system and should be consistent, should be available, should be like retrievable whenever asked by the authority, right? Okay. With this, we come to the end of this presentation. Hope you have liked this presentation. Kindly subscribe to the channel Prof Talks to get more information onto the pharmaceutical quality system. Thank you.